Hello there, and welcome to In the PI Seat, the podcast where we discuss the transition from postdoc to independent researcher and the challenges of this new role. I am your host, Camila Valenzuela, and today we have Thibaut Brunet, a G5 group leader at Institut Pasteur in Paris. Welcome to the show.
Hi, excited to be here.
I will first like to say that your model organism is absolutely amazing. I think it's pronounced guanoflagellates.
That's correct.
Okay, perfect. And for the listeners that are not familiar with this very particular model, can you describe it briefly?
Yes, so basically, our motivation is that we were interested in the very beginning of animal evolution, and in particular, we are interested in a very big transition, which was the switch from unicellular organisms to multicellular organisms. And so basically, that's the origin of multicellularity. It's also the origin of embryonic development. We researched this topic by actually looking at the group of species that are the closest living relatives of all animals. So all animals, from sponges to jellyfish to elephants to humans, descend from a single common ancestor. And we know that last common ancestor was already a fairly complex organism. It was already multicellular, it already had diverse cell types, it already had a controlled shape, it went through morphogenesis in its development. And so we are essentially interested in how that complexity evolved, so before the last common animal ancestor. And to do that, we actually look at the closest living relatives of animals, and it turns out that it's a group of single-celled eukaryotes, which are called the choanoflagellates. And so they live in aquatic environments all over the world. You find choanoflagellates everywhere, in rivers, in lakes, in the ocean. You almost certainly have eaten choanoflagellates if you've ever swam in the ocean by accident. And I said they're single cell, but in fact, that's not always completely true. In some conditions, some choanoflagellate species can develop into colonies. And those colonies they have different shapes depending on the species. But some of them resemble, to some extent, early animal embryos. Some of them are capable of collective behavior. That's the case of one of the species we study in the lab. And basically, we're interested in really understanding. So my lab's interest is morphogenesis. So we're interested in understanding how the shape of these colonies is controlled and how it emerges from the shape of the cells and from the interaction between cells. So that's basically the what captivates us in a nutshell.
That's pretty nice. Yeah, they're really. Now that you said that, I probably ate some of them by swimming.
Statistically,
Then
yes, yeah.
wonderful. I think there's a statistic that says that we eat like one spider like every few years while sleeping.
Oh
So
yeah, we swallow eight spiders in our
yeah,
life, I think. Yeah.
wonderful.
Yeah, I think you can. Yeah,
Yeah.
I decided to just swallow eight spiders at once. That way I'm done for the rest of my life.
Or then you will have 16.
That's possible.
Yeah. Okay. I would like to discuss first your general career pathway. So you're French. You were
Yeah,
born
nobody's
in
perfect.
Dijon.
Yes,
I'm so you were born in Dijon, but then you came to Paris for your undergrad. So you did your undergrad in biology,
that's correct.
right? Perfect. But then for the PhD you moved to Germany to the EMBL.
Mm-hmm.
What was the motivation for moving to Germany to Heidelberg?
It was really science, so it was a little bit serendipitous actually. And I was very lucky when I was an undergrad at the École Normale that each student had a mentor or a tutor who was like, a faculty member who was basically advising them or advising us. And my mentor was somebody named Renaud de Rosa, and he was a researcher in evolutionary developmental biology. And the reason I was matched with him was that I was really, even as a student, even actually ever since high school, I was fascinated with evolution. That was really a lifelong passion in a way. And anyway, so I was matched with him, and I asked him for a good thing about the curriculum at the ENS is that we were encouraged to do a lot of research internships in different labs. And I asked Renaud for advice on labs I could, do internships in. And he gave me a list of PIs he thought were interesting, labs he thought were interesting. And one of the names on that list was Detlef Arendt, whom I didn't really know by name at the time. Actually, I think I had been exposed to some of his work, but I didn't really know him as a person. And So one of the on that, so he had a lab in EMBL in Heidelberg, and so I just went through the list of suggested names, and I read Detlef's papers, and I was absolutely amazed by how deep and thoughtful they were. Like they really were at a very, they were really very captivating, both conceptually and also experimentally. And so I was very curious to apply to his lab. So I applied to his lab for a master's one internship, and I spent 6 months there in my master's one, and that was a fascinating environment. I mean, the MBL is a very unique place. You know, it's a little bit like a scientific oasis in the middle of a mountain in Germany. It's a somewhat unique context, and I assume many people have. If you get a chance to visit it, if only for a conference, it's really a place I would encourage people to see. And so it was very interesting, and also the intellectual atmosphere in the lab was fascinating, and there was a mixture of very rigorous, in-depth science, and also broad curiosity for the natural world and exploratory spirits that I really liked. And so after the end of the internship, I basically knew it was a place where I could go back, where I would like to go back, if possible, for my PhD. And it also helped, of course, that I knew where I was going, that I already had a somewhat extended experience there. So, and that's how I ended up applying then to the EMBL PhD program when a few years later.
But then did you immediately go to a postdoc after your PhD?
Yes. So after my PhD, I spent actually 5 years in EMBL. So I spent 3 years and a half as a PhD student and something like a few months as a postdoc to finish things, but still in Detlef's lab. And then I moved to a postdoc in UC Berkeley to actually start working with choanoflagellates. That's how I switched to choanos.
Yeah, no, that's pretty nice. And your motivation to go to Berkeley was again because of the science, or you were like looking forward to a different environment in terms of science, because it's very different, the mentality between the US and the Europe.
Yeah. It was really motivated by the science. So in a way, it's hard to optimize for many parameters at once. And in my case, I really was motivated by things I found intriguing. And at the end of my PhD, basically, so my PhD was focusing on reconstituting the evolution of muscle cells. So basically, the cells that contract and mediate movement in diverse animals. And when starting to write my thesis, and also when reading more broadly about the topic, I ended up feeling fascinated by the way it all started, by the way the control of shape and movement had emerged at the beginning of animal evolution. And one thing I was struck by from just looking at the literature or mining even public databases like the NCBI database for genomic sequences was that a lot of contractility genes that are fundamental for muscle cell function are actually found in the single cell relatives of animals. And some of them are actually very ancient, like myosin 2. You even find it in amoebae. So when an amoeba crawls, it's the same myosin 2, more or less, that you use to contract your muscles. But some others that are more specialized evolved later, and in particular, many transcription factors key for muscle cell specification evolved shortly before animals split from their closest unicellular relatives. So you find homologs of muscle genes in a way in the genomes of, for example, choanoflagellates. And I was also fascinated by reading so that made me very intrigued about choanos, and I read quite of the morphological literature about choanos when I was a PhD student, really out of curiosity. And I was really fascinated by the fact that there was a huge wealth of morphological knowledge about their behavior, about their lifestyle that had been accumulated ever since people had first seen choanos in the 19th centuries, and then which had kept, being explored more in depth over the 20th century, but largely from, just an observational standpoint with the microscopes that were available at the time, so either light microscopes or electron microscopes. And it was clear that these organisms, even though they were unicellular, had complex behaviors, that they could explore their environment, that they had a lot of sensory modalities, that they could control their shape, and that a lot of the structures involved looked tantalizingly comparable to some of the fundamental structures of animal cells. But a lot of those observations had been, purely morphological. And I also knew that there was one lab in UC Berkeley in California which had actually propelled choanos into the world of modern cell biology and molecular biology, which was the lab of Nicole King. So they had published also a lot of nice papers over the year. And what was clear about also Nicole King's lab is that it was also very exploratory. Like a lot of the lab's work revolved around understanding how coenos became multicellular, so how they switched to this colony forming behavior that I mentioned earlier. But some of it had to do with other things, like for example, how sexual reproduction was controlled. That's something also that they had described only shortly before. So it looked like an interesting lab to me in the sense that it looked like it was at the intersection of the model organisms I had ended up being fascinated by for evolutionary reasons, and also of the techniques that would allow to study these problems from a new angle. It also looked to me like the phenomena I was interested in, which was cell contractility, were not really being researched in coenos. So it looked like an open direction to research that I could maybe develop if Nicole King was open to it. And so that was the motivation. And so it happened that the lab was in Berkeley, and it happens that Berkeley is a fascinating place. But I think I've been lucky, in that sense, that it could have been anywhere else, and I probably would have tried to go there anyway. But it ended up being in California, and that also was turned out to be a place I loved. Yeah.
That's nice. Did you ever consider staying in the US and applying for a position there?
Yes. It was. It ended up being a choice at the end of my postdoc. Between staying in the US and trying to come back to Europe, and in a way, that choice was maybe more consequential than the choice of PhD and postdoc, because I was applying at the time for faculty positions. And I was, in the perspective of going somewhere I could settle on the long term. And so it ended up being a balance, of cultural factors, personal factors, closeness to friends, closeness to family. And overall, the sum of these factors clearly favored Europe. It even favored Paris, I would say, specifically. But after 5 years in the Bay Area, the San Francisco Bay Area, where Berkeley is, it also ended up feeling a lot like home. I mean, it's a fascinating place in terms of nature, culture, science. So I was a little bit split, but it felt like all other things being equal, Europe was more interesting. Another factor was that my postdoc coincided exactly with the Trump presidency. When I arrived, Trump was elected a few months after. And that was not the best time for scientists, and also, for immigrants in the US. And I was still among the immigrants who had it the easiest, but it was still stressful to even just be a postdoc, because we felt that we were not completely sure to be able to stay in the US as we would have under the previous presidency. And when I applied for positions, it was still the end of the Trump presidency. And that also favored Europe. That seemed like a safer place in that sense. After I signed to go back to France, actually, Biden was elected shortly after. So it was an interesting feeling of closing the loop, in a way, that I had arrived and Trump had immediately been elected. And I decided to leave, and Trump immediately lost the re-election. But that essentially was the way it ended up being for me. But I also really loved the scientific culture in Berkeley, and that's also a place where I could have seen myself living, I think.
That's very interesting. In the end, how we have to balance, really, how, as you said, the different factors. Perhaps we can now go back a little bit in time and talk about the beginnings of your scientific curiosity, or where did you start with science. So I would like to know, what was your first approach to science?
So the big scientific influence in my childhood was Jurassic Park. So I was born in 1987. Jurassic Park came out in 1993. So I was 6 years old. So I was really of the generation that saw it at the age where you're the likeliest to be impressed. I think the most impressionable. And that it was a big Hollywood movie, but in a way it was a Hollywood movie that tried to actually be pretty faithful to the science. There was this segment in the beginning that was explaining the very fanciful, idea, the very fanciful conceit in the movie by which they clone dinosaurs. And there was a character called Mr. DNA who was explaining, in like a short educational video that visitors of the park were watching before actually visiting, and seeing the dinosaurs, explaining what DNA was and how they had harvested it from a mosquito trapped in amber, and how they had reconstituted a dinosaur genome from that. And that's of course very fanciful, but it tried to be as realistic as it could, given the knowledge of the time, I would say. And some of the characters in the movie were also scientists, right? I mean, somewhat unusually, so some of them were paleontologists. Somewhat unusually, one of the main characters was a mathematician. It was this chaos theorist. And that, sparked a very conventional, probably childhood passion for dinosaurs. And I remember there was this magazine that was simply called Dinosaurs that was coming out every week. And in each issue, you had one bone. And if you kept collecting the issues, you could assemble a T-Rex skeleton at the end. On that magazine, actually, I looked at it again recently, and I'm pretty impressed by the level of technical depth and accuracy that it had. It was really good science popularization. Like, it really went in quite some detail for something that was aimed for children. So I think I was just really lucky that there was this moment in popular culture that was where some part of science had a really high profile, and I ended up being fascinated by that. And that evolved into a fascination with biology in general over the years. And I think I've always kept it. And when I was a teenager, I discovered the books by Stephen Jay Gould, who was this famous paleontologist who wrote collections of essays about biological evolution, which are extremely well written, extremely easy to read, but also extremely thoughtful. And I still read them with joy now. And that also was a very big intellectual influence in showing me what biology was like and evolutionary biology was like. So I think that's essentially the way it went for me.
That's amazing. I was also quite struck by Jurassic Park when it came out. I was born around the same time as you. I remember for many, many years that I wanted to be a paleontologist. In the end, I went for something completely different, but yeah, I
Yeah,
remember.
paleontology is really a gateway. Gateway.
Yeah. I think so, definitely. When you went to Berkeley, I want to know more details about that. Was it a big lab or more like a medium-sized lab?
It was about, it fluctuated, it was, I think, probably medium sized. It fluctuated between, 8 and 15 people, depending on your interns and students and so on.
Okay, so it was still quite a large lab. How was the transition? Because when you go from like having a postdoc position in a lab to having your own lab, that usually it starts smaller than the one you were in before. How was the transition of like not only transition in terms of the role that you play, but also the people that is working on the same project?
Yeah, so it is interesting because, I did both my PhD and my postdoc in relatively big established labs. And both Detlef and Nicole were fairly busy and often traveling, and the lab was already fairly well established and productive for many years. So they were not at the bench anymore, for example. And I never, for that reason, really directly see what it was like to be a new PI. Like, I never worked with somebody who was setting up their lab. I was in labs that were already, in a way, well functioning and established. And when starting my lab, in a way, I'm in the position of doing something that I never had a direct role model for, which is to play this intermediate role where you still at the bench, you still have to train people at least. No one else can teach people how to work with choanoflagellates. I have to do it. Even though now it's getting better, but in the beginning I really was the only person who could do it. And also, I'm still doing experiments. I actually want to be able to stay at the bench for a few years if I can, because to some extent I like doing it, and I think I can still be useful there. But it is a little bit, it feels intermediate, like being between being a postdoc and being a fully established PI who often is further from the bench, even though not always. Some PIs manage to stay at the bench until the end of their career. But I think it comes at a cost. It takes quite an effort to be able to do that. So in a sense, it's it feels a bit like a weird hybrid between the PI job I've seen and the postdoc job I've had. I think what helped a bit is that as a postdoc, I had supervised quite a few students. So one interesting thing about the American system is that PhD students very often do rotations. So their first year, they spent working in, they do maybe 3 months in 3 different labs, and then that way they can choose which lab makes more sense for them as a match. And so I supervised quite a lot of rotation students, including one of them who ended up joining Nicole King's lab, and with whom we had a really, really good collaboration. And that already, and I supervised also undergrads. I also, when I was a postdoc, I worked very closely with two grad students, so those with whom we discovered C. flexa, the Coenoflagellate species that much of my lab works on right now. So it was a very close collaboration, and these were already grad students who had a few years of experience, so I was not training them from scratch, but it still felt like not working entirely on my own already. And so in that sense, I had some experience in supervising students or working at least closely with students when I was a postdoc, and that was felt like it was very much reactivated when I started my own lab. So that helped the transition being smooth. But I'm really glad I had that earlier experience. Otherwise, it would have been really, really, really tough.
Yeah, for sure. I think even like starting to mentor students at the very beginning, it's kind of a daunting task. But then it is different when you mentor students now versus what you did as a postdoc. Because now, for example, you have to hire people, like you have to choose them. How do you approach those kind of things?
Yeah, so that's a really good question. So I've been, I've discussed a lot with some of my senior colleagues in Pasteur when I started the lab, and everybody has been extremely helpful on giving a lot of advice. One of the pieces of advice I was given was to not try to grow too fast, but actually hire people one at a time. Take time, you make sure you take time to properly train them and mentor them. And also, of course, make sure that you find a good match. When you start the lab and you only have a few people, you have to make sure these are the right people, right? And so my approach has been, I think, fairly conventional in the sense that I really take time to know people before recruiting them. For students, ideally, they spend an internship in the lab before committing to a PhD, which is exactly what I did, before committing to my PhD in Detlef's lab. For postdocs, I really try to interview them very much in depth. So they invited on campus, they give a presentation. We have very long scientific discussion. They also meet all the members of the lab one-to-one. They have individual discussions. They often come for lunch or dinner or both. And I check references, of course, very, very carefully. So in a sense, I try to really get a full sense, not only scientifically, but also from a human standpoint of people I recruit, and to maximize information, make sure there is compatibility between not only me and them, but also the existing lab members and them. So that's something that I actually was very inspired by the recruitment process of my postdoc mentor, who was very extensive in doing that and in paying attention to the vibe of the lab and to make sure that new recruits would be a good fit. So that's the way I'm, and also, it's of course fair on the other side. Like postdoc candidates, PhD candidates have to make sure they know me, that they know the lab, that we're compatible, that we can work together, that we're excited by the same things. And so that's the way I've been doing it so far. And so far I've been extremely happy with the people I've recruited. I feel sometimes incredulous that they chose to come to a new lab, and I feel very lucky. But I think that's the way I've been doing it, being very slow and careful and as human as possible about it.
That's nice. Do you think setting up expectations at the beginning, like during the interview process, it plays a role in it? Like really being direct with what you expect from the people and what they can expect from you?
Yeah, for sure. I think it's important that they know a lot of things about our research. One of the constraints is that coenos are not easy organisms to work with. So I always tell them that we are in this trade-off where there are a lot of very interesting open questions, but there are still relatively few tools, even though we have more and more. And that it requires quite a lot of both persistence and creativity to solve problems. We don't have a huge community to rely on, even though there is a small Coeno community, which is very, very collaborative and altruistic. I think we're also lucky in this respect. But it is important, that people know the style of research that we do, that they know the style of mentorship they will receive, and that they know it's a good match for them. It's not a good match for everyone. There are all kinds of personalities in science, and there are all kinds of research that match the different kinds of personalities. So yes, absolutely, it's important that it works both ways, right? I was saying maximizing information on both sides. People should know what they should expect.
That's good to hear. You started in Pasteur in 2021.
That's right.
So far, I mean, it's only been like less than 2 years, but I don't know if you had to deal with issues and if they are different from what you had to deal with as a postdoc.
Yeah, I mean, for sure, the job is extremely different than a postdoc. When you're a postdoc, you're essentially a researcher, and you apply a little bit for funding, and you manage also a little bit people because you can supervise students. But when you're a PI, you suddenly become a researcher and an accountant and a manager and to some extent a salesperson. You have to sell your research to funding body. You have to present it in conferences. You become a recruiter. You become also responsible for, like the equipment in the lab, the repairs. You have to interface with the technical services. You have to interface with the service maintenance from the different providers. You have to buy equipment. You have to decide what equipment to buy. So the number of tasks you have to do becomes multiplied manifold. And of course, everybody is only human. And it's very unlikely that you will enjoy all of these tasks. It's very unlikely that you will be good at all of them. So you do have to find a way to, do the best job possible at all, balance the time between all, get advice for the tasks that you're less naturally good at or that you've maybe never practiced before. And so one big challenge is to also plan your time, because you have so many directions when you start a lab and all of them are important, but you cannot do all of them at once. And it depends a bit on personality, but for example, I'm not very good at switching between tasks. I'm like diving deep into a task and then focusing on it for a few hours, ideally uninterrupted, and then maybe switching to another. And that's a challenge actually, to work in that way when you have a lot of different things to do, when also there is a lot of random encounters, people coming to your office, people calling you on the phone, asking you to deal with that or that thing. So planning the time around the diversity of things you have to do, and sometimes knowing how to protect the time where you really have to focus, that was a challenge. And I'm still learning how to be a PI, I think. So I'm learning how to do it. But that's something I really discovered as a PI. As a postdoc, the diversity of tasks, I say, was smaller and more manageable without any explicit organization. Now it requires a bit more advanced planning.
So we already discussed with Nicolas the G5 position basically, but I would like to have your perspective on why this is a particular and an interesting position to apply to.
Yeah, so it's essentially it's a PI position. So in France, a lot of research positions are staff scientist positions, where you join the CNRS or the INSERM, and often you would join a pre-existing team, and that's different than maybe more American type PI position, where you really start a new team from scratch. In my case, it felt like the right match because I was studying something that was relatively new in France, which was, studying choanoflagellates. So it made sense to start a new team because of the topic and because of the openness of the questions. It was really largely motivated in my case, again, by the science I was interested in doing. If I had done a different type of research, it maybe would have made more sense to apply as a chargé de recherche in a pre-existing team. And I think I could also have liked that. I mean, it comes with maybe less administrative burden at first, even though I'm sure chargé de recherche also have a lot of stuff to deal with. Yeah, I thought the G5 position in Pasteur looked like a very interesting position because it matched the kind of research I wanted to do. And it comes with, the kind of support that you get from other big institutes across Europe and across the world, which is that you get some startup package to actually recruit people and buy equipment and pay for consumables. And you don't get that in many other French institutes. You get that to some extent you get that, of course, in, for example, in the EMBL, in the Max Planck in Germany, in the Crick Institute also in the UK, for example, or in many American institutes. But in France, Pasteur is I think, in a somewhat rare situation that it can offer that. It also comes with a lot of support from colleagues. So we have, again, a matchmaking system with senior colleagues who can act as mentors. There was a lot of help I received from directors of department when I started, from other G5 colleagues who had started a few years earlier, from more senior colleagues. I feel like I've really been very fortunate to be so supported. It also comes with administrative support, actually. So we have an administrative assistant, which we share with 3 other labs. And that's extremely precious to have somebody actually who is an expert in paperwork. And I think it would have been much, much harder without the support of that person. Her name is Rizlan. And also the G5 position, another nice thing which I think is relatively new is that we have a support group for new group leaders. So the new G5s who started within a bracket of maybe 2 years, we meet regularly either with somebody from administration or even by ourselves to exchange tips and to discuss our experience. So Nico Raskovan is one of my colleagues in my cohort. And that actually was very, very helpful. Had actually a lot of very useful discussions with Nico about how to hire when I started, and he really helped me a lot. So yeah, overall, I think that's an interesting position. It comes with a challenge, of building a new team and switching to independence. In my case, directly after the postdoc. It's not the absolute best position in the world. I think it depends on the research you want to do. I think in my situation, it was the best position I could apply to and the best I would get. And yeah, I'm very, very happy I'm here now.
It's very good to know that there are like very supportive positions within institutions. So, in terms of like the different positions that are available, did you apply to anything else, either in France or elsewhere in Europe?
Yeah, I was fairly open-minded actually between, being a PI or being a staff scientist. I was not close to it, even though, of course, I would have needed to find a lab that was ready to accept choanoflagellates. Or being even a university lecturer, like I was also considering applying for maître de conférence in France. I started with the PI jobs because of the calendar. I think like a lot of offers were coming up, and it was not yet the recruiting season for CNRS or for maître de conférence. So I started with applying to PI jobs, and because I got the one in Pasteur, I then did not apply to the other positions. But I was actually very open to different things. And so yes, I applied to a few others. Again, because I got Pasteur pretty quickly, I didn't apply to so many others. But I applied to the MBL again, and so I was interviewed, and that was actually a super interesting interview process. I did not get the position, but I'm still glad I applied, if only because the interview was so interesting, and we can talk a bit more about it. And I also applied to another position, which was in Lyon, the Institut de Génomique Fonctionnelle, which is a smaller institute with something between 10 and 20 labs, I think, which is focused on really organismal biology and understanding organisms in an integrative way from the genome to, like the physiology and the evolution. And that was a very intellectually dynamic environment, a smaller institute than Pasteur, but also a place where it seemed there were great colleagues. So that one I also would have been very interested in going, I think. But I ended up choosing Paris and Pasteur in part because of the G5 position, in part also because for personal reasons I liked more the idea of living in Paris. So in total, I think I only applied to 3 places, but also because I got this one quickly and I knew it was my first choice if I got it, so I accepted it. But I would have applied to many others otherwise.
Yeah, I was going to ask if this one was your first choice. That's nice. I'm intrigued now about the differences in the interviews for EMBL and Pasteur.
Yeah. So actually, yeah, the interview in the MBL it has a somewhat unique format in the sense that they organize a symposium. So all the candidates actually come to campus at the same time. And so we all visited the MBL with a cohort of something like 6 other maybe candidates. And we got to see each other's research presentation. We got to go to dinner all together and with the committee. And that's something that I don't know if Pasteur does that actually usually, but when I interviewed for Pasteur, it was already during COVID, so it was online. So I did not get to see the other G5 candidates, right? But so the MBL symposium format I thought was super interesting. Like I met very, very interesting other junior researchers, and I'm still in touch with, I think pretty much all of them actually now. Yeah, it was a very, very very nice experience in that sense. And psychologically also it deflected a bit the pressure of doing a job interview. I could have the feeling I was going to an interesting symposium. Yeah, that I think was a very nice experience. In Pasteur, it was constrained also by COVID, right? I did everything by video from California because I was not physically allowed to. I would have been physically allowed to go to France, but I would not have been allowed to re-enter the US. There were still very strong restrictions. So I had to do everything in early morning because of the time difference, interviewing from either from my living room or from the lab in Berkeley. So that was a challenge, but it was I still followed the strategy of trying to maximize information, talking with as many colleagues as possible, trying to understand what it was like in Pasteur. And in the end, I felt I could get a good sense of what the institute was like, even without having visited it.
Is it a bit unusual for, I feel, ANS scientists to not do an internship in Pasteur when they are
Yes,
younger?
that's interesting. I don't know actually. I don't know the statistics.
Yeah.
It's I think I didn't do so many internships, right? I did like two or three, and so I did with three actually. I did one in Oxford, and then one in Curie, and then one in the EMBL.
Mm.
So statistically, I think it's a small sample. So the fact that Pasteur is not in it,
Yeah.
I think it's just random. But yeah, that's true. I could have done an internship in Pasteur and I didn't. But I think I was very focused on evolutionary biology, and there were not so many evolutionary labs in Pasteur maybe at the time. I think now there are more, not only mine, but quite a few others.
Yeah, probably the field changed a bit inside the institute. My last real question is, what has been the most rewarding experience during this whole transition for you?
Yeah, it's hard to know, because there are so many different kinds of experience, right? I mean, there's a human side, of course, where you interact with people and you share excitement, and that's a very important part of what keeps me going. There's a scientific side where you are more in a dialogue with nature, and you try to trick nature into revealing its secrets, and it's extremely exciting when you unlock something, and that also is an important part of what keeps me going. I think one very striking memory is the discovery of C. flexa, which is this coenoflagellate species that we co-discovered with some colleagues from Berkeley back when I was a postdoc. And so it was basically at the time a new species of coenoflagellates, which we found essentially by chance, actually. And so the way the story goes is that after my second year of postdoc, we went to a field trip to Curacao, which is an island of the Caribbean. And we went there with Nicole King and with two grad students, Ben Larson and Tess Linden. And we went there really for, without having a clear research plan in mind. We went there because there was a workshop on protistology, so the study of free living single cell eukaryotes, or actually single cell eukaryotes in general, that happened there. And we were interested in going there to exchange techniques with some colleagues. And as I said, there are coenoflagellates everywhere in all aquatic environments. So we were interested also in simply sampling across Curacao to look for what kind of coenoflagellates we would find there. But there was no reason to expect anything particularly unusual, I would say, at first. It happened that during a tour of the island, we collected water from a splash pool so that these are basically pools that are on the shore, so very close to the ocean, and which are fed by the waves. So they're not tide pools, but they're splash pools. And we collected a sample of water there, but without really again expecting anything in particular. And it was not even a place where we went for scientific sampling motivations. We went there because we were touring the island, and we happened to go there at some point. And so we brought those samples back to the marine biology station, which is in Curacao. And and it was actually a stepwise discovery. First we found some small coenoflagellates that were shaped in chain colonies, and these are basically lines of cells, files of cells that are swimming around. And we were very excited to see those because we were not completely sure this had ever been seen in nature. We knew that some of our coenos were forming them in the lab, but we were not sure that they had been described directly in a wild sample before. And actually, maybe they had, but we just didn't know. And just seeing them was exciting. So it was a motivation to go back to that site. And then we went back, and I remember it was an evening pretty late. We looked at samples from those same splash pools. So we collected many others. We saw the first thing we noticed were coenoflagellate colonies that were shaped like trees. And basically, so these were very, very, very large colonies which were branched, and at the tip of each branch was one coenoflagellate. But it was clear that the entire rest of the tree, the entire structure, the branches and the trunk, this was all extracellular matrix which had been secreted by the coenos somehow. And those trees had a very, very beautiful geometric shape. They were different than other tree-shaped coeno colonies that we had seen before. And so we were extremely excited about seeing those. And we were actually interrupted in this observation by the fact that there was a barbecue dinner with all the people who attending the workshop that we had to go to. And so we went there, and we basically, I think, annoyed everyone by telling them, oh, we found those trees, aren't they incredible? And I think no one was as excited about them as we were, but we were like, okay. And then we came back after the barbecue, so it was pretty late, and we were not drunk, but we were we were in a cheerful mood after a long night, and I think it was already something like midnight, and we went back to the lab to look a little bit more at the samples. So this was with the 2 grad students. So we were already in a strange mood, because it was late at night and it was this unusual context, and we wanted to just see if we could find more trees. And then we found yet another species of interesting organism. And what we could see was that it was a colony of microbes that formed something like a monolayer of cells, so something like a sheet, a bit like an epithelium, but completely detached from any organism, free swimming. And we could see that this colony of organisms could deform very quickly. So a little bit like an animal, like it looked like it had a very exploratory behavior. And it was shifting shapes in front of our eyes. And by looking more closely, we thought it was maybe even a colony of choanoflagellates. So actually the group we were interested in, but we were not sure because it was a wild sample, and you get all kinds of things in those samples, of course, all kinds of organisms. So it was a super strange discovery moment of seeing this species that didn't look like anything we expected to see, that looked super interesting scientifically, and also seeing it in this very, very strange context, being back late at night alone in the lab without anyone to show it to. Nicole King had already returned to Berkeley at the time. So I think we were lucky to be able to make movies, to actually record like those very early observations, because we thought, if this organism doesn't survive in culture, if we lose it, and if we admit that we only saw it once super late at night, nobody will believe us. And I think it was a great moment. First because of the completely serendipitous finding, the super unusual context, but also because it was a shared moment with two PhD students who I was working with at the time, and we ended up collaborating, on the description of this species. We are 3 co-first authors on this on the paper that described this species. So we then were lucky, and I think that it was a lot of hard work, but also a part of luck that we could put it in culture and then study it in the lab. And we eventually named it C. flexa, so we had to name the species. But we are 3 co-first authors on the paper that describe it, and I think I was also very lucky that we were all, I hope, lucky that we ended up being those 3 people in particular, because we worked extremely well together. We all had a shared enthusiasm for describing this organism and understanding how it worked. All very had, I think, we could all partake in each other's scientific interests and share work in a way that was collegial and collaborative, and there was never even an ounce of competition between us. It was always very mutually supportive. And so, that I think inevitably stands out as an experience, both as a discovery and also as a collegial interaction.
That's nice. Yeah, I think that really highlights how the team you're working with it's really an essential part of science. Everyone, like in the general population, they keep thinking of scientists as like this kind of like almost like a hermit in a basement working on their stuff. But actually, I think it's a really social thing, like how we really push each other to do better things when we work as a team.
Yeah,
So I think that's really interesting.
Yeah, I think so. And I think
Yeah.
in this case, there was a great complementarity of perspectives and expertises. So Ben Larson he still is a physicist. So he had a great intuition, actually, I think, for the geometry and the physics of the system. And Tess and myself, we were evolutionary biologists, but we also had different backgrounds. She had done quite some population genetics as a grad student, and I was more coming from the microevolution side. And because we are looking at a completely new organism, I think all intuitions and all perspectives ended up being very complementary and very, very useful to really approach it from all kinds of different angles and characterize it in an integrative way. And I think the interaction was more than the sum of its parts, and that's been the case very often in my career, actually. Like out of, I think I have 5 first author papers from PhD and postdoc and also internships, and 3 of them are actually co-first author papers, and I've always really enjoyed a lot working with, as an equal with colleagues and bouncing ideas back and forth, and it's always been much more than the sum of its parts. And for sure, this aspect of science, that we do science also as a group and also for the community, we also do it to share findings that others will be excited by and build upon. That's a very important dimension.
I think that's a nice note to end the conversation today. So thank you so much, Thibaut, for taking the time to talk with me. I'm going to give you the chance now to plug anything you want, projects, conferences.
Yes, so I mean, for people who are interested in choanoflagellates, there is every few years a choanoflagellate workshop, and we're working to organize one within a few months. And it's a great way for the community to stay in touch and and get to know each other and support each other, investigating new model organisms. And it's not only open to people who work on co-NOs, but also often on other close unicellular relatives of animals.
Yeah, that's super nice. So thank you so much again, and hope everyone else enjoyed the conversation, and we'll be back in the next episode.
And that's the show, folks. If you enjoyed listening, then hit that subscribe button to get the latest updates straight to wherever it is you're listening. Don't forget to rate us on Spotify or Apple, and follow us on Twitter at MotionPod. You can find links to things we've just discussed on our website, preprintinmotion.com. If you'd like to tell us what you think, then send an email, shout at us on Twitter, or shout at us if you see us walking down the street. This has been a JMJ production, generously supported by our friends at ASAP Bio. Until next time, have a good week.